Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
This website makes learning so easy. I found all my resources here.
Like what you share.
We analyze diverse genomic architectures, exploring human cytogenetics and pathologies to resolve complex aberrant chromosome systems.
Eukaryotic systems feature complex, linear chromosomes in prokaryotes. Human cytogenetics reveals various numerical pathologies and distinct, aberrant structural formations. Eukaryotic genomes are packaged into chromatin. The DNA helix winds around an octamer of core histone proteins. This fiber is organized into distinct topological domains.
Chromatin is classified into active euchromatin, and highly condensed, silent, heterochromative regions remain permanently condensed, while facultative domains undergo dynamic changes.
The diploid human genome consists of 23 pairs. This includes autosomes and sex chromosomes. Physical mapping reveals size and gene density. Aberrations lead to clinical phenotypes. Chromosome 1 is our largest chromosome. It contains over 2,000 coding genes. Mutations here lead to Gaucher disease, and therapies include enzyme replacement. It encodes 753 protein codes include HTT, linked to Huntington disease. Clinical management focuses on symptomatic care.
Over 181 million base pairs, encoding critical genes like SMN1. Mutations cause spinal muscular atrophy. Gene therapies offer new management hope.
135 million base pairs. It contains heme-coding genes, including insulin. Mutations cause sickle cell anemia and beta thalassemia. Management includes transfusions, CRISPR gene therapies.
Chromosome 15 spans over 100 million base pairs. It contains 613 protein G genes. Mutations cause Tay-Sachs and include supportive and dietary care. Chromosome 17. It contains over 83 million base pairs. Key genes include mutations can cause breast cancer disease. Management includes PARP inhibitors and physical therapy. Chromosome 21 spans over 46 million base pairs. It contains 231 linked to Alzheimer's and Down syndrome. Management includes early screenings and targeted care.
Chromosome X is vital, spanning over 156 million base pairs. It contains over 800 protein-coding genes. Mutations cause conditions like hemophilia.
Chromosome Y, with over 57 million base pairs, contains key genes like SRY. Mutations can cause azoospermia. Management includes assisted reproduction.
Chromosomal abnormalities are divided into numerical and structural anomalies. Aneuploidies are primarily caused by meiotic non-homologous chromosomes fail to segregate properly during gametogenesis.
Trisomy 21 is the most common. Trisomies 18 and 13 are more severe. Sex chromosome aneuploidies are less severe. This is due to X inactivation. Microdeletion and microduplication syndromes result from structural anomalies during meiosis. They disrupt key gene dosages, leading to distinct phenotypes. Diagnostics relied on karyotyping. Modern diagnostics have transitioned to molecular cytogenics, including FISH, microarrays, enabling detection down to the single nucleotide level.
Precise chromosome segregation is essential. Centromeres function as the assembly site for kinetochores. These mechanisms vary between mitosis and meiosis, is conserved across all eukaryotic life.
Conversely, some species possess holocentric chromosomes, which lack a single primary construction. Instead, they're kinetric sister chromatidyl to a spindle pole.
A chromosome break causes major issues. In monocentric organisms, fragments are lost, but holocentric fragments segregate properly and survive.
While holocentricity provides stability during meiosis, reciprocal crossovers link homologous chromosomes. Cohesion along the chromosome arms is cleaved, allowagotosegraded.
In C. elegans, a single crossover occurs. This divides the bivalent into long and short arms. while cohesin is protected along the long arm. This prevents chromosome breakage. Notably, this meiotic segregation is completely independent of SE and SEMP-C, demonstrating a functional uncoupling of meiotic kinetochore assembly from mitotic centromeric chromanin. Inverted meiosis flips the rulebook of cellular division. Here, sister chromatids separate in the first round, leaving homologs connected by thin chromanin threads.
Understanding chromosome dynamics requires analysis. Primary association arises from sequence-specific pairing. These are stabilized by chiasmata at pachytene. This determines regular segregation at metaphase I. Secondary association occurs at pro-metaphase. It is driven by biochemical affinity, indicating ancestral genomic homologies. Some cells develop unique, giant chromosomes. They support high levels of gene expression. Visible puffs indicate active transcription. These are master templates.
Chromomeres appear as beads along the chromosome axis. They serve as anchors from which lateral loops emerge. These loops are sites of active gene expression.
First observed in amphibian oocytes, they consist of two homologous chromosomes with symmetrical lateral loops.
These lateral loops are coated with an asymmetrical matrix. Transcription progresses from a thin end to a thick end. This allows RNA polymerase II to access the DNA template at very high rates. Polytene chromosomes display alternating dark bands and light interbands. The dark bands are highly condensed heterochromatic regions.
Specific bands expand into puffs and Balbiani rings. These puffs represent uncoiled, active DNA-mRNA synthesis. This enables high-volume protein synthesis.
Beyond standard definitions of scale, mini-chromosomes and microchromosomes coexist with chromids, redefining genomic complexity.
Mini-chromosomes are small, functional chromosomes. They replicate in eukaryotic cells. In prokaryotes, they contain essential genes and are formally classified as chromids.
Microchromosomes are tiny. Chicken genomes have 30 pairs. They are incredibly gene-dense. This is vital for avian evolution. Analysis reveals distinct properties, separating macro and microchromosomes. Macrochromosomes are typically larger, while microchromosomes are smaller but highly gene-dense.
Microchromosomes are ancient, stable elements. In mammals, they fuse to form large macrochromosomes. Their high gene density keeps avian genomes compact, supporting flight. Bacterial genomes were once thought to have just one chromosome, yet many contain large secondary replicon systems. These essential elements are called chromids. To be classified as a chromid, a replicon must use a plasmid-type system, have a nucleotide composition close to the chromosome, and carry core essential genes typically found there. Chromids differ from standard plasmids. Plasmids carry non-essential accessory genes. True chromosomes use DNA-mediated replication initiation. Replication of the CAR-TS site on chromosome 1 triggers the initiation of chromosome 2 by binding to RCB, ensuring both cycles terminate synchronously.
Vibrio cholerae has two chromosomes. R1 uses DA at ORI1. R2 uses RTB at iterons to initiate, but binds 39 mers to repress.
Bacterial secondary chromosomes evolve more rapidly. They show faster substitution rates than primary ones. Analysis reveals weaker codon usage bias. This drives faster evolution. This fast evolutionary rate has two main drivers. Secondary chromosomes replicate later, reducing gene expression. They act as testbeds for new traits. This helps bacteria adapt and survive anywhere.
Genomes host atypical chromosomes. These parasitic elements ignore standard rules. They drive cancer and chaotic rearrangements. These are the genome's ultimate parasites.
Neochromosomes are giant, abnormal marker chromosomes, found in approximately 3% of all human cancers. They contain hundreds of amplified DNA fragments.
Dozens of double-stranded DNA fragments are produced. Lacking telomeres, circular structures replicate. Unequal crossovers generate dicentric rings. Spindle forces pull them to opposite poles. This drives random genomic amplification. Breakage fusion bridge cycles generate massive copy number gains. Key oncogenes like M2 and CDK4 are amplified. Unbeneficial passenger genes are epigenetically silenced. BFB cycles erode the centromere. This drives the cell into a mitotic crisis. A neocentromere forms to resolve it. Capturing telomeric caps fixes the marker.
B chromosomes are genomic parasites. They do not pair with standard chromosomes. They use chromosome drive to accumulate. This drive breaks Mendelian rules.
B chromosomes arise from standard A chromosomes. They rapidly accumulate repetitive elements. This triggers an arms race with the host genome, where suppressors evolve to limit their drive. In jewel wasps, the PSR chromosome tricks the system. It eliminates paternal genes, leaving only itself and the maternal set. This converts a diploid female egg into a haploid male, ensuring its own transmission.
In Lake Victoria cichlids, B chromosomes determine female sex. They carry a dominant female locus, overriding the standard XY system, driving transmission.
Sex determination is surprisingly flexible. In many species, temperatures shape this process. This occurs through environmental epigenetic plasticity.
In humans, sex is fixed at fertilization. The XY system is the most common. The SRY gene on the Y chromosome acts as the master switch. It triggers testes development.
Under ESD, sex is established after fertilization. It is triggered by non-genetic environmental cues, such as nutrients and light.
During the long days of early season, young crustaceans develop as males. A longer growing season grants them larger size, which directly correlates with mating success. Settle on the seafloor and you become a large living female, but land on a female's proboscis and you'll become a microscopic male, living inside her as a parasite.
For slipper snails, position is everything. When the base female dies, the male above her changes sex to take her place.
In many reptiles, sex is not determined by chromosomes, but by the incubation temperature. Crucial development happens in the middle third, known as the thermosensitive period.
Reptilian TSD-IA, or MF pattern, means low temperatures produce males, while high temperatures produce females.
In some reptiles, by temperature, low temperatures produce 100% female offspring, while high temperatures produce 100% males. This is seen in tuataras and some lizards. In alligators, eggs incubated at extreme temperatures develop as females, while intermediate temperatures produce males.
Wise reptiles use temperature for sex determination. Selection favors the fitness of one sex over the other. Warmer nests make larger females, maximizing egg production.
TSD is regulated at the molecular level. Warm temperatures upregulate the scene. Aromatase converts androgen. Cool temperatures keep aromatase low. Exogenous estrogens force sex reversal.
Temperature signals alter DNA methylation. Male temperatures activate the DMRT1 gene. Female temperatures silence it to activate FOXL2. This establishes the sexual pathway.
Daphnia show how ESD transitions to GSD. Chromosome 1's locus overrides plasticity. Suppressed recombination isolates the DFH chromosome evolution. We must contextualize the human paradigm. This matrix compares chromosomal parameters. We map genetic functions across diverse species.
Gallus features a complex genome. It has macro and micro chromosomes. Defects cause severe immunodeficiency. Management requires genomic selection.
Caenorhabditis elegans features holocentric chromosomes. Specific genes like HIM3 are vital for meiotic arrest. Defects cause meiotic non-disjunction, managed by RNAi studies. Vibrio cholerae is a pathogenic bacterium. It features multi-replicon circular chromosomes. Key genes like RCB initiate its replication. Cardi's deletion causes lethal cycle arrest.
Meet Nasonia vitripennis, the jewel wasp. Specific PSR locus disrupts RUP's paternal chromatin. This drives complete disternal genome. Manage maternal modifiers to neutralize it.
Astatilapia latiflid fish. It has monocentric and chromosomes. Key genes, like brain aromatase, are duplicated and threaten survival. Therapies and Hyshin manage these risks.
Is an actinobacterium and a chromid. Key clusters direct spore pigment and surfactants. Mutations halt stopping spore formation. Plasmids and surfactants restore development. Nature uses diverse chromosomal configurations. These balance stability and adaptability, from rigid human control to dynamic plasticity, spanning bacterial chromids and B chromosomes.